Professor Brinton here with episode 8 of the Note Card Review. Our topic is ploidy and some of the differences between meiosis as opposed to mitosis. We're going to start with ploidy. Ploidy represents the number of copies of a particular chromosome found in an individual. So if we have this cell here as one of your own cells, or a simplified version of it, you can see the two different chromosomes, 1 and 2, in this particular cell in prophase that's headed into mitosis or meiosis. For each of these two chromosomes, you see two copies containing the same basic information. These are homologous chromosomes. This is the maternal and paternal version of a particular trait. So if this is the gene tied to your eye color, okay, that is going to be found on all of these in the same location, simply the maternal version and the paternal version of that gene for eye color. You also have sister chromatids and non-sister chromatids that are essential to understanding the steps in meiosis and mitosis. These two on one version, on say the maternal version of a chromosome, one homolog, those are the sister chromatids. The other two versions, if we're comparing the different homologs, those are non-sister chromatids. So if we compare this piece of our first version, say the maternal one, and so this one and this one, these are non-sister chromatids. So they still contain the same basic information, but it's a maternal and a paternal version as opposed to both maternal or both paternal on the same chromosome. So we have a homologous pair of chromosomes, sister chromatids within one version, non-sister chromatids if we're comparing the two different versions, so the two different alleles, for example. Now, to connect that to mitosis and meiosis, ploidy is going to change with meiosis. It is not going to change with mitosis. So if you go through mitosis from beginning to end, you will have the same ploidy, the same number of copies of each chromosome at the beginning as you do in the daughter cells. If you go through meiosis 1, you will reduce the number of copies of chromosomes by half. So you'll go from whatever cell you have to begin with to a haploid cell in meiosis 1 every single time. This is reductional division. That's the reductional portion of meiosis. If you instead look at meiosis 2, the second half, it is going to look very much like mitosis. So as you proceed, the ploidy is not going to change. At the beginning of meiosis 2 and the end of meiosis 2, ploidy is the same, just like in mitosis. To look at the broader picture, we need to compare interphase versus the M phase. Both versions of the cell cycle, meiosis and mitosis, are going to have interphase. Why? Because the cell still needs to grow. It still needs to replicate the DNA. So in both cases, you will have the G1, S, and G2 portions of the cycle. That's not going to change. What will change is what happens during the M part of the cycle. If we're looking at mitosis, you'll simply have one copy or one round of prophase, metaphase, anaphase, telophase, and cytokinesis. If instead you are looking at meiosis, then you're going to have two copies of the entire cycle, going through meiosis 1 and meiosis 2. So this is why you have telophase actually occurring 3 times. You split the first pair of cells in half in meiosis 1, and then you split both of those cells in half again during meiosis 2. So you have every single stage, prophase, metaphase, anaphase, telophase, and cytokinesis, occurring during both phases.